Okay, hello girls. This is your tutorial for how to calibrate a spec and start this experiment. So the first thing you need to do is to turn on the spectrophotometer. I turn mine on to get it kind of warmed up, but the way you turn it on is by turning this knob, let me make sure I'm looking down, turning this knob on the far left. You turn that up, okay, and your machine will turn on. The next thing you need to do is adjust the wavelength. The wavelength should be set for you, but you just want to double check that it's set to 470 nanometers. So you can turn this knob to adjust the wavelength. And the wavelength is pictured right here. The next thing we need to do is make sure we are measuring in absorbance. So we want to click this button so that it now reads absorbance. Once we have this turned on and you let it warm up for about 10 minutes, this one was warming up prior to starting this video, we need to calibrate our machine. We do this using a blank. So I have a couple of different test tubes in front of me. I have an E tube, that's the enzyme tube, the substrate tube, the B or the blank, and then the reaction cuvette. So we're going to use our blank cuvette. These little tiny test tubes are called cuvettes, and they're made so that they can be used for this machine. They make the glass very accurate, so that way the glass doesn't actually interfere with the absorbance. So what we're going to do, we need to first clean off any oils or anything on the outside of this. You'll make this blank by combining water, guaiacol, and the peroxidase enzyme, everything but substrate. You want to clean this up really well, and then you want to place it in here. Okay, this is where the sample will be read. So we're going to place it in close. Oh, let me do that so you can actually see. Okay, we're going to place the sample in here, close the lid. It's very, very important that we keep the lid closed to get an accurate measurement. Then we want to adjust the absorbance using this knob right down here. You can see that the reading for absorbance is right here. We want to zero this. So we're going to turn the knob to get the absorbance reading down to 0. Get it as close to 0 as you possibly can. You'll be pros at this by the time you actually do your experiment. And every time you do a run, you need to recalibrate the machine. So you always want to make sure that you are taking it back to 0. Okay, so once we calibrated it, we're going to remove our blank and we want to start our reaction. Okay. Sorry, these glasses are moving a little bit on my face. Okay, so once we do that, we need to combine our substrate and our enzyme tube. So the substrate tube contains hydrogen peroxide and water, and the enzyme tube contains peroxidase, the enzyme from the turnips, guaiacol, and it is going to contain water to make the volume 7.5 milliliters. This is a correction from your lab. The guaiacol should be in the enzyme tube. Okay, so that way, because hydrogen peroxide naturally decomposes and we don't want to measure any of that. So what we're going to do is combine our enzyme and our substrate tube together. Okay, this is going to be very quick, so you want to make sure that you work as a team and start the timer. Because I'm doing this by myself, I may have a slight delay. Okay, so combine the enzyme and the substrate tube together. You want to make sure you get everything mixed really well. You have parafilm to help you mix this. Start the timer as soon as you can. You need to get a reading at about 30 seconds, so you need to get this in the spec in 30 seconds. Okay, once you do that, pour it into your reaction tube. You will have extra, so pour it as close to the top as possible. You want to clean it. Make sure it's clean. And get it inside here to measure it. Okay, I am very close to 30 seconds right now, and I would take my first reading. You can measure the absorbance right here at 30 seconds. It's like 0.09. Now I'm going to let this continue to go. I'm going to take a recording at 1 minute. I'm going to take a recording at 1.5, 2 minutes, 2.5, and finally at 3 minutes. Okay, every time you're going to be measuring the absorbance of the reaction. This is very good to do as a group, so that way you can make sure you get the timing down. But generally speaking, this is how you're going to run the reaction, and we will review this very quickly at the beginning of class on Wednesday.